Welcome students. Today we are going to discuss leaf abscission and senescence. First, we will discuss abscission. In the normal course of development, plants routinely shed organs such as leaves and fruits. This may be part of a normal aging process or a response to stress. Aging is a process by which maturity is attained with the passage of time. Physiological changes by which juvenile plant is converted to a mature plant, and the gradual decline in vigor and loss of viability of stored seeds are examples of aging. While aging is a natural expression of the slowly accumulating changes that make an organism more vulnerable to disaster, leading ultimately to death. The term abscission is derived from the Latin abscindere, meaning to tear. Shedding leaves in one way to reduce the surface area for transpiration in times of water stress, or to prepare a deciduous plant for an impending winter. The process of shedding organs is known as abscission. Abscission occurs as a result of a development of a special layer of cells called the abscission layer near the base of the petiole. As the organ ages, the cell wall in the abscission layer weaken and eventually separate. The abscission zone consists of one or more layers of thin-walled parenchyma cells resulting from anticlinal divisions across the petiole. In some species, these cells are formed even before the leaf is mature. Just before abscission, the middle lamella between certain cells in the distal region of the abscission layer is often digested. Abscission is achieved through the induction of genes that code for cell wall hydrolytic enzymes. For example, specific cellulase genes are induced in abscission zone in association with the abscission process. In bean, a specific cellulase isoform is induced at the separation layer, but also in a less localized pattern in adjacent vascular tissue. This digestion often involves synthesis of polysaccharides, hydrolyzing enzymes, most importantly cellulose and pectinases, and their secretion from the cytoplasm into the wall. Formation of these enzymes is accompanied by a rapid rise in respiration in cells of the proximal part of the abscission zone. The rise is similar to that which occurs in climacteric fruits and also involves increase in polyribosomes, characteristic of cell actively synthesizing proteins. Furthermore, one or more layers of these proximal cells increases in size while the cells of the abscission zone distal to the breaking point do not. These wall digestion processes accompanied by pressures resulting from unequal growth in expanding proximal and senescent distal cells of the zone apparently cause a break between them. Ethylene causes cell expansion and secretion of cell wall degrading hydrolases in some species. Several environmental factors promote abscission, especially drought and nitrogen deficiency, but these first hasten senescence. Even though abscission is an active process in cells proximal to the break, the organ being shed nearly always first becomes senescent. Now, role of hormones in abscission. Abscission appears to be dependent on the relative concentrations of auxin on either side of the abscission layer. In an intact plant, abscission appears to be dependent on the direction of auxin movement within the petiole and ultimately on the competing effects of the leaf blade and the shoot apex. The auxin content of a young, rapidly growing leaf is relatively high, decline as the leaf ages and approaches senescence. Abscission is prevented as long as the leaf continues to release auxin. As the leaf ages, the release of auxin declines and abscission is stimulated. If the leaf is removed, abscission of the petiole is also accelerated. Similarly, removal of the shoot apex delays abscission of the petiole, but if auxin is applied to the cut stump, the effect of an intact shoot apex on leaf abscission is restored. Now, we will discuss senescence. Every autumn, people who live in temperate regions can enjoy the beautiful color changes that precede the loss of leaves from the deciduous trees. The leaves change color because changing day length and cooling temperatures trigger developmental processes that lead to leaf senescence and death. Senescence is not simply death or necrosis, which could be caused by poisons or other external factors. Rather, senescence refers to an active developmental process that is controlled by the plant's own genetic program. Senescence is the age-dependent deterioration process at the cellular, tissue, organ, or organism level, leading to death or end of lifespan. Leaf senescence is not a passive process and unregulated degenerative process. During senescence, leaf cells undergo rather orderly changes in cell structure, metabolism, and gene expression. The earliest and the most significant change in the cell structure is the breakdown of chloroplast. Metabolically, carbon assimilation is replaced by catabolism of chlorophyll and macromolecules such as proteins, membrane lipids, and RNA. Increased catabolic activity is responsible for converting the cellular materials accumulated during the growth phase of leaf into exportable nutrients that are supplied to developing seeds or to other growing organs. Thus, although leaf senescence is a deleterious process for the sake of the leaf organ, it can be seen as an altruistic process. It critically contributes to the fitness of whole plant by ensuring optimal production of offspring and better survival of plants in their given temporal and spatial niches. Leaf senescence is thus an evolutionary selected developmental process and comprises an important phase in the plant life cycle. In agricultural aspects, however, leaf senescence may limit yield in crop plants by limiting the growth phase and may also cause post-harvest spoilage such as leaf yellowing and nutrient loss in vegetable crops. Thus, studying leaf senescence will not only enhance our understanding of a fundamental biological process, but also may provide means to control leaf senescence to improve agricultural traits of crop plants. In naturally senescing leaves, senescence occurs in a coordinated manner at the whole leaf level. usually starting from the tips or the margins of a leaf towards the base of a leaf. However, when the uneven environmental stress is targeted locally on a leaf, the stressed leaf region undergoes earlier senescence than do the other parts. Thus, leaf cells show some degree of locality in a senescence program. Leaf senescence can occur without an obvious correlation with senescence of other organs in some plants, such as many tree species, although it is often developmentally coordinated with senescence of other organs or whole plants, especially monocopic plants. Now, we will discuss types of senescence. Senescence occurs in variety of organs and in response to many different cues. Many annual plants, including major crop plants such as wheat, maize, and soybean, abruptly yellow and die following fruit production, even under optimal growing conditions. Senescence of an entire plant after a single reproductive cycle is called monocopic senescence. Other types of senescence include: number 1, senescence of aerial shoots in herbaceous perennials; number 2, seasonal leaf senescence as in deciduous trees; number 3, sequential leaf senescence in which the leaves die when they reach a certain age; number 4, senescence of dry fruits; number 5, senescence of fleshy fruits; number 6, senescence of storage cotyledons and floral organs; and number 7, senescence of specialized cell types, for example, trichomes, trichetes, and vessel elements. Now, patterns of senescence. Senescence in plants shows a wide range of patterns. Number 1, cellular senescence; number 2, tissue senescence; number 3, organ senescence; number 4, organism senescence or whole plant senescence. Number 1, cellular senescence. Individual cells or a small number of cells of an organism may undergo senescence while the others do not. Both gametogenesis and embryo development involve orderly sequences of cellular senescence. During female gametophyte formation, 3 out of 4 cells produced by meiosis degenerate. The synergids and the suspensor cells also sense during embryo growth. Number 2, tissue senescence. In many circumstances, large group of cells or tissue may degenerate and die. The tapetum, that is the layer of cells that surround the microsporocyte cells or pollens, degenerate, while the nuclear layer next to the growing embryo may also break down. The senescence of groups of cells in particular locations may play a role in leaf morphogenesis. Number 3, organ senescence. Even a single organ may follow diverse patterns of senescence, which is exemplified by leaves. Leaf senescence may be progressive. The older leaves sense more or less at a time. Floral senescence may also be synchronous on a seasonal basis. Even within a leaf, the cells may sense at different times and rates. Some leaves sense from the apex towards the base, while senescence in others starts over the main veins, showing vein yellowing and then continues in the intervenal regions. The senescence of leaves and branches in response to environmental stress represents an adaptation either to reduce the water evaporative surface or to allow better light perception by the plant. Number 4. Organism senescence or whole plant senescence. Whole plant senescence is accompanied or preceded by a decrease or cessation of both shoot and root growth. The reduction in vegetative growth occurring during the reproductive phase have been recognized for a long time in both monocopic and polycopic plants, where the shoot apex is converted to an inflorescence as in corn and xanthium. Node production ceases coincident with the appearance of flower bud. Now, what are the physiological changes that occur during senescence? The entire physiological processes decline during senescence and a multitude of changes lead to death. In this respect, most of the biochemical work deals with changes occurring in the leaves either as detached organs or attached in whole plants. The effect of senescence on different metabolic processes are: number 1, photosynthesis. It is generally observed that the net photosynthetic rate increases as leaves grow and then declines gradually at the time of maximum leaf expansion. This indicates that the rate of photosynthesis is a function of individual leaf age. During the rapid phase of whole plant senescence, young and old leaves degenerate almost together. The rate of photosynthesis also declines with equal rapidity in young and old leaves. Simultaneous senescence of leaves occurs not only in monocopic senescence but also in autumnal senescence polycarpic plants. Although loss of chlorophyll content is a commonly used index of senescence, as with chlorophyll content, the synthesis and activities of chloroplast enzymes, for example, ribulose 1,5-bisphosphate carboxylase, decline after the cessation of leaf growth, which is parallel to the loss of photosynthetic activity. Calvin cycle enzymes that are synthesized on chloroplast ribosomes decline earlier during senescence than those synthesized primarily on cytoplasmic ribosomes. Number 2, respiration. The respiratory apparatus of senescing tissue remain active until late in senescence and then decline rapidly. Loss of respiratory capacity can be a factor in the final stage of senescence, whereas there is evidence that the initial stage of senescence is not linked with respiratory decline. The mitochondria often swell and decrease in number, but the changes occur late in senescence. Amino acids have been found to accumulate, giving rise to a change in the respiratory quotient. A kilometric rise in respiration may occur in the senescence of both intact and detached leaves. From the fact that respiration decreases only late in senescence and that the lack of oxygen supply prevents the breakdown of chlorophyll and protein, it appears that cellular energy is required during senescence, possibly for the synthesis of the degradative enzymes. The respiratory kilometric of senescing leaves is correlated with a rise in RNA levels and an increase in protease activity. Number 3, nitrogen fixation and mineral uptake. It is interesting to note that depodding, which increases the availability of photosynthate, can cause appreciable increase in nitrogen fixation and delay nodule senescence. In a similar way, the decline in whole plant photosynthesis parallels the decline in nitrogen fixation. Symbiotic nitrogen fixation decreases with the aging and senescence of leguminous plants. Such decline in nitrogen fixation is associated with the structural changes of individual nodules and bacteroids, which is followed by their senescence. Root growth as well as root function usually slow early in the reproductive phase. Just like nitrogen fixation, mineral uptake through the roots suffer a decline during the reproductive phase. Number 4. Protein and nucleic acids. The most basic of all events accompanying senescence is the decline in protein and nucleic acid levels. It has been shown that although there is a decline in protein, RNA, and DNA of senescing tissue, but these three metabolites do not decline at the same rate. There is an early gradual decline in protein and RNA about the time when vegetative growth ceases, while DNA decreases last. The initial decrease in protein and nucleic acids in the chloroplast involves a decrease in the major chloroplast enzyme, ribulose 1,5-bisphosphate carboxylase, and the other enzymes which are synthesized on the chloroplast ribosomes. The loss of these enzymes is correlated with reduced ability of the chloroplast to synthesize protein and RNA, coupled with a reduction in the number of chloroplast polysomes. Number 5. Membranes and organelles. Of all the cell organelles, the chloroplast shows the earliest symptoms of physiological decline. As pointed out earlier, decrease in ribulose 1,5-bisphosphate carboxylase activity is noticed first with a corresponding early loss of chloroplast ribosomes, and this precedes the decrease in cytoplasmic ribosomes. In later phases of senescence, however, pronounced ultrastructure changes in the thylakoids occur. Other changes occurring at this time include swelling, vasculation, disappearance of endoplasmic reticulum and Golgi bodies, together with the loss of endoplasmic reticulum associated ribosomes. The galactolipids and sulfolipids decrease before the phospholipids that predominate in the non-chloroplast membranes, suggesting that the biochemical decline may occur relatively early in chloroplasts. Although the changes in chloroplasts occur early, they remain intact till the time when the tonoplast ruptures. As soon as the tonoplast disintegrates, the hydrolytic enzymes, acids and toxic compounds are released from the vacuole, and this triggers the cellular degradation. In the final stage of cellular degradation, the plasmalemma disintegrates and the nucleus undergoes massive alteration, whereas the mitochondria may persist with intact shape even at this stage. It is generally acknowledged that membranes seem to play an important role in plant senescence. Now, we will discuss a relationship of environmental factors and leaf senescence. Our current understanding of the relationship between environmental responses and leaf senescence mostly comes from the study of senescence response to the phytohormones such as abscisic acid, jasmonic acid, ethylene, and salicylic acid that are extensively involved in response to various abiotic and biotic stresses. These stresses affect synthesis and or signaling pathways of the hormones to eventually trigger expression of stress responsive genes, which in turn appears to affect leaf senescence. The roles of these hormones in regulating leaf senescence are becoming evident through characterization of genetic mutants and the global gene expression analysis, providing important molecular information about how the hormonal signaling pathways lead to changes in pattern of gene expression during leaf senescence. The well-known senescence promoting hormones are ethylene, abscisic acid, jasmonic acid, and salicylic acid, whereas senescence retarding hormones include cytokinins, auxins, and gibberellins. Dear students, this is all about leaf abscission and senescence. Thank you very much.